The aim of this experiment is to demonstrate the visualization of in vivo protein dynamics and localization patterns in living Drosophila embryos by time-lapse confocal microscopy. This is accomplished by first harvesting embryos from maintained Drosophila stocks. Next, transfer the embryos onto a slide and then manually remove the chorions from the embryos. Dechorionated embryos are subsequently mounted in an organised fashion onto previously prepared slides, and, depending on the experiment, may be micro-injected with appropriate spindle checkpoint-inducing reagents. The final step in this procedure is to perform time-lapse imaging of embryos using confocal microscopy. Ultimately, time-lapse confocal microscopy is used to show spindle checkpoint protein co-localization, chromosome behaviours, and the effect of checkpoint-inducing agents in checkpoint-related transgenic Drosophila lines.
The method can help answer the key questions in the mitotic regulation field, such as the effect of the mutations or depletions of spindle checkpoint protein through the use of the transgenic lines on the co-localization of the other checkpoint components, and the consequence of this on the ability of the spindle assembly checkpoint to function efficiently.
Transgenic flies are maintained at 25 degrees Celsius in plastic vials containing fly food and with additional dry yeast powder on the top. The vial is routinely replaced every 2 days.